17.6 is looking at gene expression among the different forms of life, prokaryotic and eukaryotic. What we need to understand before we move forward is that genes are universal. They are occurring in all living things. In some cases, and in many cases actually, the same genes are found in different organisms. Or if you take a gene from one organism, you can transplant it into a different organism. And that's because DNA is universal as well. So, archaea are prokaryotes, but they share many features of gene expression with eukaryotes. Bacteria and eukarya differ in their RNA polymerases. They also differ in their termination of transcription of ribosomes, and archaea tend to reassemble eukarya in these respects. Bacteria can simultaneously transcribe and translate the same gene. We looked at why they can do that previously, and that's because there is no nucleus. So the mRNA that is transcribed from the DNA is going directly to the ribosome because they're both found in the cytoplasm or cytosol of that bacteria. However, in eukarya, transcription and translation are separated because the transcription process occurs within the nucleus. Therefore, they're bound by this nuclear envelope, which is the, so to speak, skin of the nucleus. Translation, on the other hand, that's out in the cytosol. Also, archaea, transcription and translation are likely coupled. Once again, they are prokaryotic organisms, so therefore they do not have the nucleus either. So archaea would be happening simultaneously. So looking at that coupled process as it occurs in prokaryotic organisms, we can see that we have some RNA polymerases which are transcribing the mRNA, but we also have ribosomes attached to the same strand as well. So almost immediately after that mRNA is popping off of the DNA, ribosomes attached to it, forming that polyribosome, many ribosomes, and they immediately start translating the growing mRNA. So there's not really any modifications that happen to the mRNA of prokaryotic organisms. They don't have that opportunity to splice out any introns, so to speak. So revisiting genes. Genes, they are unifying concepts of life. There are certain ways that you can consider genes. Discrete unit of inheritance, region of a specific nucleotide on a chromosome, DNA sequence for a specific polypeptide. There's different ways you can look at a gene, but regardless, you have to consider all of these things when you're talking about a gene. So there isn't really one simple definition to what a gene actually is, because it's all these things. So this is tied to question 81. I'm skipping over 80, which I'll come back to. This is showing us an overview of chapter 17, the transcription and translation, the last two components of central dogma. Keep in mind, central dogma involves replication as well as transcription and translation. Question 81 is asking you to put the entire chapter together of transcription and translation, which I'm going to do here. I'm going to discuss that. But first, you might want to pause this, label in some of the components from the screen to your image in your handout if you'd like. And I am going to kind of go through this kind of as an overview. I'm not going to be including every single little detail, although you're going to need to know a lot of that stuff. So don't think just because it's not mentioned here that you're not responsible for knowing it. Things like RNA polymerase, DNA polymerase, the couple different forms of RNA or DNA polymerases, that sort of thing. So what we can look at here is we see transcription, which is where we're using a template strand of DNA to produce an mRNA strand, which is called a primary transcript. The primary transcript will be modified before it leaves the nucleus. This would obviously be in a eukaryotic organism. And it will snip out the introns, leaving the exons. So the exons, it looks like, are these deep red colored things, and the introns would be the lighter red color. So the introns are removed, which shortens the mRNA, and it's only the exons that are expressed in the finalized product of the protein. But before this can leave the nucleus, we have to pop on a poly A cap, as well as a 5' cap, which is actually, you can't see it until you get down here. With these caps, that does a couple different functions. One is it allows the RNA to now leave the nucleus. The RNA cannot leave the nucleus without these caps. The caps also serve to protect the genetic material within the preserved exons. So there's a second function of the 5' cap and the poly A tail. And also it allows the ribosome to pick up an mRNA. Remember, the ribosome has a bonding site, which is illustrated right here, which is where the cap will actually bind to allow us to ka-chunk our way through the process of translation. So mRNA goes into the cytoplasm, and all at the same time we have the large and small subunits of a ribosome combining together. They'll form those hydrogen bonds. The mRNA will lock in place, and then we can start going through the process of translation. So we'll use the codons and anticodons to pull in the tRNA molecules to bind to the codon. It will lock in place starting at the AUG or start codon, which is actually seen right there. Once that's locked in place, this tRNA will be shifted to the P site, and that will expose the A site. We can have another tRNA molecule come and bind in place based off the anticodon-codon pairing. We'll form a peptide bond between the two amino acids, and once that bond is formed, the bond between the tRNA and the amino acid is broken, which is why there's no amino acid on this one, and we'll shift over to the E site. This will shift to the P site. This will open up for a new tRNA to come in. That was called translocation. This tRNA molecule, once it has reached the E site, will be released. It's no longer bound to the mRNA, and the tRNA will go to one of the synthetases, which will reattach an amino acid to it, allowing it to go back to the A site of a ribosome. Once this is all done, we reach the stop codon. We add in some water, which makes this all fall apart, and we have our large and small subunits go floating off from the cytosol until it's time to repeat. The mRNA, it could exist for 2-4 more weeks, or it could get broken down pretty immediately. It varies on the type of mRNA, but it could be reused is the point. It could be reused. Not necessarily is, but it could be. So in a nutshell, that is what we have been talking about in Chapter 17. So going back to question 80, question 80 is asking us to summarize what a gene is. And we started off with the one gene, one enzyme theory, which as we saw is true to an extent, but there's so many ways to describe a gene that it makes it rather difficult to actually use that as a true and accurate definition, although that's the most widely known one. So in summary, a gene can be defined as a region of DNA. That region of DNA can be expressed to produce a final functional product, and that's either going to be a polypeptide or an RNA molecule. So that's question 80. Let's take a look at some of the things that you should know simply for review processes. I'm going to put up a few pictures on my slide here, and I'd like you to look at those, and just for review, you don't need to turn this in or anything like that. Think about what's going on in these pictures. Think about some of the vocabulary that would be coined with it. So as I go to this image, what are we looking at here? What process is this? What are some of the key terms that you maybe should be thinking about when you see an image like this? If you'd like to partake, you might want to pause the screen and just think about it, maybe go back, review some of your notes. Here's the next image that you could take a look at. Once again, what's going on? What are some of the key terms? A third image, what do we see happening? What's going on? What are the key terms? What is it tied to? Some different sort of practice. Can you fill this chart in? Are these 4 questions and 2 questions on the next slide something that you could discuss? So here's the first 4, and here are the next 2. Keep in mind, this stuff I am not looking for. You do not need to turn this in. This would simply be self-review, but what I would be looking for was the 7 or 8 multiple choice questions on, I believe it was page 350, as well as, I want to say it was question 39 of the reading outline, chapter 17 specifically. And yeah, it was question 39. I'd be looking for those multiple choice questions and number 39 from the reading outline to be turned in for an assignment grade by Thursday, which is when we're taking our Unit 6 test. You can either do this on a separate sheet of paper and turn that into the turn-in box in my classroom, or you could do it on your reading outline and then take a picture of that and email that to me. Either way is fine. I have no preference. If you have any questions over anything that we've been looking at, shoot me an email, and I'll be checking my email periodically throughout the course of this weekend.